Hello, in this video we will discuss about the skeletal muscle, the anatomy and physiology we will understand in this video, so remember. So please watch the full video to understand the physiology and anatomy of the skeletal muscle. And what is the skeletal muscle? Skeletal muscle is voluntary muscle we can move with our will. It means the voluntary means we can control it, their movement. So this is a bone which that is attached with the tendon, and tendon a tendon attached with the muscle. So the bone to muscle attachment is through tendon, while the muscle to muscle or bone to bone attachment is known as a ligament. So this is the tendon and epimysium are present. So the skeletal muscle controlled by the motor nerve and get the reflexes. So reflexes mean the motor nerves through after the sensation of the something like the pain, and after this sensation will go into the nervous system, the spinal cord, into the brain, and brain will transmit the signal through motor neuron to contract the muscle. So this is the fascicle and the perimysium are present inside, and the fascicle contain this muscle contain many fascicle. This one fascicle contain here is the one fascicle contain blood vessel and the wrapped by perimysium, and this is basically the endomysium is the inside is the between the individual muscle fiber. So inside is basically fascicle contain many muscle fiber. So remember the fascicle contain many muscle fiber, and it contain a myoglobin, which that is oxygen stored protein. So oxygen will reserve for the surplus of the oxygen, while on the other hand also the creatine phosphate are present for the extra energy preservation. So this is the muscle fiber, this muscle fiber, and this is basically the myofibril contain the muscle fiber contain myofibril. So the muscle fiber is the unicell, basically is the cell of the muscle. It is also known as muscle fiber is the myocyte. So the muscle fiber, let's begin to understand the muscle fiber. It contains a nucleus, so the multinuclei. So the more than one nucleus are present, and inside is the myofibril. And this myofibril contain many sarcomeres. So remember this is the myofibril, and the dark band and light band show in the diagram. While this is the orange color is the mitochondria will provide the energy continuously, and the sarcoplasm are present is the outer region, while this is the transverse tubule or T tubule are present, which that is used for the movement of the calcium ion. We will discuss in the next diagram, but remember just this is the and sarcoplasmic reticulum are present. The sarcoplasmic reticulum is used for the storage of calcium, and on the time of requirement the calcium will release and bind with the myofibril, the troponin. So okay, and this is the light band and dark band we will discuss now. So this is the myofibril. This myofibril contain a many sarcomere. So what is sarcomere? Sarcomere is the unit of muscle contraction. Here you can see the H zone. This is the H zone, and the M line are present, and this is the I band, the isotropic band. Isotropic band mean the light can pass, while the A band mean the anisotropic band contain thick filament is myosin, while the black color shows the thin filament is the actin. And the actin is used for the and myosin is used for the muscle contraction. That is, here you can see the contracted muscle will bind with the actin with myosin head. So this myosin is the protein and actin also protein. And relaxed state, we can say this is the cross section of the sarcomere, one sarcomere, and the relaxed stage is this. So this is the sarcomere. This is the unit of muscle contraction. We are understanding now the single sarcomere. Z to Z line, between the Z to Z line, the sarcomere is, and the M line between the sarcomere, while the actin filament is the thin filament, while myosin filament is the thick filament. So this is the Z line, Z disc are present, which that is attached the titin. The black color shows the titin. Titin will bind with the myosin with the Z disc. So the A band and I band, which that is showing in the diagram, while the troponin is the attached with the thin filament, not thick. But this troponin is used for the binding of the calcium when calcium will release from the sarcoplasmic reticulum. So this is the A band and I band. I band mean isotropic, A band mean anisotropic, mean the light cannot be passed from the A band. And this is the myosin head, is used for the attachment with the actin, and the actin myosin bridge formation through when troponin will bind with the calcium will allow to move the sarcomere, and contraction will lead to decrease the length of the sarcomere. So let's begin to understand the step. So the active site on actin is exposed as calcium binds troponin. After the calcium will bind with the troponin will lead to activate. The myosin will bind. After the myosin will bind, the ADP and the phosphate are present already. So the actin and tropomyosin are present is the thread-like structure, and bead-like structure is the actin. So anyhow, this is the myosin head. Myosin head in the form of pair, which that will be bind, and the junction formation is known as the actin tropomyosin bridge formation. And the second step is the myosin head forms across bridge with actin, and in this the ADP is already present. While this is the bridge formation due to the troponin will bind with the calcium, and the third is the during the power stroke, myosin head bends, and ADP and inorganic phosphate are released. After the releasing of the inorganic phosphate, the ADP will be separate and the ATP will be added. A new molecule of ATP attached to the myosin head, causing the cross bridge to detach. So the detachment is very important. The ATP after the death, the ATP will be reduced, ATP will be decreased, will lead to contraction of muscle impossible, is known as the rigor mortis. So here is the ADP again formation, the calcium will be released, again move into the sarcoplasmic reticulum, while the detachment will occur and the cycle will begin for the again contraction. So the step 4 is the ATP hydrolyzed to ADP, and the phosphate which return the myosin to the coaked positive. And in this way, let's begin to understand the action potential, how it work, how the calcium will bind. So remember, this is the motor nerves. The motor neuron is control the muscle contraction, and in this way the neuromuscular junction is present. This neuromuscular junction basically is the synaptic knob of the neuron. The motor neuron contain vesicle. This vesicle contain basically is the neurotransmitter acetylcholine. The acetylcholine receptor are present in the sarcolemma. So this is the sarcolemma, mean the muscle fiber, and in this way the acetylcholine is the neurotransmitter will release is known as the exocytosis, and the synapse, the neuromuscular junction is present. So the acetylcholine receptor will bind with the acetylcholine. After this trigger, the sodium ion will move inside. After the sodium ion will move inside, this is the T tubule. T tubule will the depolarization will move the T tubule side, and the DHPR receptor like channel will allow to calcium move inside into the sarcoplasmic reticulum, and where the clastobin 1 protein as well as the PRYR ryanodine receptor 1, which that will be allowed to calcium bind, and in this way the calcium will move out, the pump out the efflux of the calcium. Calmodulin will bind for the regulation of the calcium, the maintenance of the calcium in the outside, will lead to troponin bind, and after the contraction will occur and the calcium will be moved again through a Sirca channel into the sarcoplasmic reticulum for the complete the mission. So the sarcolemma here and contain the calcium sodium calcium efflux exchange. So I hope you make sense about the skeletal muscle and the anatomy and physiology, the function of the skeletal muscle, how it will be work, and the skeletal muscle controlled by the motor nerve and get the reflexes, and it is the voluntary, not involuntary. And this is the skeletal muscle require the contraction by the helping of the bone. So it is the endoskeleton of the human being.